All right, guys. Good morning. Welcome to the digital image processing workshop. So just to give you a sense of what's going to happen in the next 3 days. So we're going to start today by kind of a lecture that mostly is going to talk about the concepts of imaging and kind of the motivation of why we're even interested in learning image processing. And then very quickly, we're going to transition away from slides and into actually doing stuff using Jupyter notebooks. So this is all in Python, this class. So we assume a certain basic knowledge of Python, but even that is not strictly necessary if you're good at Googling things. So there's going to be a lot of Googling over the next 3 days. We're going to go through what are known as pixel level operations today. Probably it's going to spill into tomorrow. We're going to talk about thresholding and image segmentation, which is the process of identifying objects in an image. And finally, we're going to talk about kind of feature extraction and getting as much information as we can out of parts of an image. And finally, we're going to finish with some intro to some more modern approaches that are kind of what's going on in computer vision in the last few years. This workshop is an introduction, so this is not going to be a very comprehensive thing. You're not going to come out of here being experts, but you're going to come out of here knowing a lot of, hopefully, the core concepts of traditional image processing. The other thing that's important for you to take away from this class is kind of why is this even necessary? Why do we want to do this in a computational way as opposed to just doing everything by clicking on screens and doing everything by eye? So hopefully this motivation would be also somehow instilled in you. Now, this is, like I said, a basic introduction. You can dig deeper if you want, and these are a few resources. Now, all of these slides are online, so don't bother writing anything down or anything like that. These are a few resources, but these are a few of many. So if you just go online and search image processing for X, it would give you a long list of options. And so following this, we're gonna just go ahead into kind of the motivation of this course. So what is an image, right? When you look at the world and you see something around you, your vision system. So what exactly is happening there? So photons are emitted or reflected from things around you. They are focused by a lens. You have a lens in your eye. And also by the cornea. So the cornea is literally doing most of the heavy lifting in terms of focusing. It focuses on the retina, which is in the back of your eye. The retina is basically a screen. It is a 2D array of photoreceptor cells, and these photoreceptor cells are, I'm simplifying here, of course, but generally speaking, are sensitive to different colors. And so this information in multiple colors, actually 3 colors, in our retina is passed to the brain via the optic nerves, and there it's processed, right? And we do our processing in our brain. So this is an image of how we see the world. Now, what is a digital image? So a digital image is very similar, honestly. So you have this lens. The lens focuses the light on a sensor. It's the same type of 2D sensor. I mean, not the same type, but very much inspired 2D sensor that collects photons, well, converts photons into electrons. Of course, computers store information in bits, so everything needs to be digitized. This is why there is this word digital in the course definition. So this is what digital means. It means everything is converted into integers, effectively. And then this information is stored as a big matrix of numbers. So from the point of view of the computer, an image is just a large matrix of numbers, right? So what exactly are each one of these pixels storing? So usually, for the most part, it stores an integer in one of these ranges. And you can imagine that different types of integers would give you different precision in terms of how many levels of light you can differentiate. So if I have 65,000 levels, I can differentiate more than if I have 255 levels. The downside of this is that it takes more to store more information, right? So the cost of higher precision is more memory cost, so more memory use. And also, sometimes it's pointless, depending on what exactly we want to get out of an image. It's not necessarily true that we want to keep as much information as possible. Okay, so like I said, an image is this set of numbers from the point of view of the computer, and when you these intensity numbers kind of map back to what we perceive as an image, right? So far, we're all good. Any questions about this? Okay, so a lot of times when we talk about imaging in any context, we talk about resolution, right? And the reason we have to talk about resolution, in a perfect world, there would be no such thing as resolution. We would always be able to resolve any two objects, no matter how close they are. We would be able to say how far they are from each other. But unfortunately, nature doesn't work that way. Light does propagate as a wave, and when waves coincide, they can be either destructive or constructive in their interference. And lenses are actually designed to maximize the constructive interference at a certain distance, which is called the focal plane. So this is what the lens does. But of course, you're losing a lot of the information because you're only collecting some of the light that's emitted. And because of that, what you actually get on the other side of a lens is not a perfect image of your source. You get some blurred image of your source, and it's blurred by something that's called the point spread function. So the point spread function is basically the image that you will get from a point source. If you take a picture of just a point, on the other side, you won't get a point. You'll get something that looks more like this Gaussian, or whatever you want to call it. Okay, so what is image resolution? So image resolution kind of depends on how much of this light you can collect. Right? So the more of this light that comes out of this point you can collect, the better your image resolution potentially would be. And this number, how much light we can collect, is called the numerical aperture. So if you've ever done any either microscopy or just used a camera, any lens comes with this number, NA, a numerical aperture, which all it is how much light can this lens collect from an object. Now, a larger numerical aperture, basically more light that can come in, would mean a thinner point spread function, and I will show in a minute how this translates into better resolution. Right, so are you more or less with me so far? Okay, so what exactly is resolution? So I've been using, what? Numerical aperture. So I've been using this word image resolution. What exactly do I mean by image resolution? So image resolution is actually motivated by physiology. So it's the question, if I have 2 points that I'm imaging, how far away can they be for me to still resolve them as 2 points? At some point, from my point of view, they would look just like one blob. So if they're right on top of each other, I would get this one blob. If I separate them a little bit, I might be able to start resolving them, but not really. It just looks like one kind of stretched out blob. At some point, I would be able to say, this is actually 2 points. And this is the point where I say, this is my resolution. And Lord Rayleigh came up with this formula for image resolution. And you see that it depends on the wavelength of the light that you're using to image and on the numerical aperture. You're going to have a different resolution in X and Y than you are going to have in Z, so the depth. And usually the resolution in Z is bad compared to the resolution in X and Y. So NA is typically, the numerical aperture is typically on the order of about 1/2. The wavelength is typically on the order of about 500 nanometers. And so you see that the resolution in X and Y is typically on the order of about 500 nanometers, right? 0.6 divided by 0.5 is roughly 1. And so 500 nanometers. Here, I'm going to have 2 divided by 0.5 squared, so 0.25. So this is going to be 4 times my wavelength, so about 2 microns. So it's going to be 4 times worse than my resolution in X and Y. Okay? This formula is also called Abbe's diffraction limit. Okay. Okay, so let's.
Sorry, what is N?
Oh, sorry. N is the refraction index of your media. The refraction index of air or vacuum is about 1. Water is about 1.3. Oils can be 1.5 to 2, depending on what you're using. Okay, so one thing that you probably noticed, or maybe not, in these formulas is that there is something that we usually care about in objectives that doesn't appear in these formulas. Can you have a guess or an idea? When someone tells you about the new objective that they got, what is the first thing they usually tell you about?
Magnification.
Magnification. As you can see, magnification does not appear here at all, right? So magnification, at least from the point of view of optical diffraction, is not a factor in how much resolution you have. But there is a certain caveat to that, which is that the size of our pixels is finite, right? And so if the size of our pixel mapped back to our image, so divided by our magnification, is larger than these numbers, then this is going to set our resolution, and we're going to just say that the system is just not diffraction limited, right? So the thing that's going to set the resolution for this image is not going to be optical diffraction. It's going to be just the physical size of the pixel and how much area they take up in image space. Okay, is that clear? More or less? You have any questions? Okay, so this is kind of the idea of what is imaging and what exactly do we mean by resolution. And the next thing is what exactly is image processing, right? So this is an image processing workshop. We're going to learn a little bit about how to do image processing, but what exactly is it? And image processing for us is a very simple task, right? We see this picture, and it's very easy for us to say, this is a bunch of monkeys. They're sitting on a rock. There is some stuff in the background. They have different sizes, different weights probably. One is browner than the other, and so on and so forth. Computer vision is the attempt to teach computers how to do what I just did very easily with almost no effort, and any person can do with very little effort, right? Okay, so what is not image processing? So image processing is not magic, right? We can't create data or create information when it's not there. If we are trying to create information when it's not there, that's called image forgery or image manipulation. Don't do that. It's always, or almost always, better to fix problems with your data on the acquisition side than on the processing side. For instance, if you ran an experiment and your sample went out of focus, it's better to repeat the experiment and make sure you're in focus for the whole time than to try and salvage it and get something out of these blurry images, right? And this is just one example, but this is generally true always. And so the usual principle of garbage in, garbage out also holds here. So having good data to start with is always key. So the next question is, why do we even need this? Because we just said that it's super easy for us to do it by eye, right? So what's the point? Maybe it's just better. These are Luddites, by the way. They broke stuff in the 1800s, machines. And so you can say, why do we need computers to do this, right? We can just do it ourselves. And so let's talk about kind of the history of optical microscopy. So before the year 2000, so BFP is before fluorescent proteins, we were mostly dealing with antibody-based labeling, so in terms of microscopy, mostly of fixed samples. We had very low throughput, so a typical experiment would be a few megabytes. You'd hold it on a floppy disk, a few tens of images. You'd go, you'd look at your images, you'd click through it, you'd segment things by eye, and everything was great, more or less. There was some problem of cherry picking, but let's say that we were good, rigorous scientists. We made sure to have controls. We double-blinded our data. We did everything right. And okay, this more or less worked back then. The problem is that in the last 20 years, there has been kind of an explosion in the scale of data that comes out of microscopy. experiments, and also from other fields. So the same is true for astronomy, the same is true for self-driving cars, the same is true for many different fields. And suddenly, things that used to work very easily, and like, I'm going to open these 10 images and click through and everything's going to be great, aren't working anymore, because instead of having 10 images, we have 10,000 images or 10 million images. And so these approaches aren't really scalable to data of that magnitude. So as soon as imaging became a big data and a high throughput problem, all of these approaches kind of went out the window. And so you saw this explosion in development of what's called computer vision. Now, in terms of reproducibility, you can make the strong argument that having algorithms that we design to do our, to process the data is a more rigorous approach than just picking things by eye, because it's less prone to cherry picking, right? I'm not going to take the one cell that did what I want to say in the paper that cells do and throw out the 99,000 other cells that don't do it, right? So I can't really, I mean, we can always cheat, but for the most part, it's harder to cherry pick and be non-systematic in the way you analyze your data. So do people have comments or questions or ideas about this? Does anyone have an anecdote, a paper that was solely built on cherry-picked data? Okay, I won't go there. We don't want to call people out. And so what computational image processing is we take this image, we do something that relates to computers to it, and we get on the other side a list of features, in this case, monkeys. Each monkey has an ID, a number, some characteristic of its sex, some characteristic of how big it is, and whatever, right? So we extract whatever features we want out of this data, and this is kind of the goal of computational image processing. Yeah. The numbers here, it's more or less random. So it's just the fact, it's just so you can keep track of, for instance, monkey number one. You and me can talk about the same monkey in the same language. So we say this one in the corner is monkey number one, and it is a female. It weighs 24 somethings, and so on and so forth. Right? But it's more or less random. So this process of just numbering individual objects is called image labeling, and we're going to go over it at some point. It is important, if you want to track stuff over time, to preserve the label. Right? So monkey number one in the first frame should be monkey number one in the second frame. Okay. Okay, so what might an image processing pipeline actually look like in practice? So we start by image acquisition, either by camera or by microscope or by telescope or by whatever mean you want to acquire your image. We're going to do some pre-processing. So often we need to align our image. Sometimes our field of view is limited, so we need to tile multiple fields of view into one larger image. Sometimes our image is noisy, so we have some some noise kind of destroying our signal. So we want sometimes to denoise or remove some speckles and stuff like that. After we did this pre-processing, we're going to do actual processing. So we're going to decide what is the foreground of our image and what is the background of our image. This is usually a very important step in almost any image analysis pipeline, right? We want to know what we care about and what we don't care about. Often we're going to then segment the foreground into objects that we care about. So in case of microscopy, these objects usually are going to be cells, but these objects can be people, they can be cars, they could be stars, they can be anything. Then we're going to extract interesting features about each one of these objects. And finally, we're going to convert this into some form of digestible data. So in the same way as we had this matrix of monkeys, right? And then we're going to do data analysis. And data analysis is not something we're going to cover in this class or this workshop for the most part. Data analysis is taking the data, so this matrix of things, and learning something from it, right? So I can, for instance, take this picture of monkeys and learn that there, in any group of monkeys, there is always one male and everyone else is a female, you know? But this is not something that is specifically an image processing task. It's something that is a data processing task, right? So any extraction of information out of this data that we generated in our image processing pipeline. So what kinds of images are we interested in? What we might encounter, and this is of course partial list. So these are craters on the moon. This is a large cat. These are MRI scans. This is an X-ray image. These are fluorescent cells, which how many? So maybe I should have done this to start with. How many of you are from like astronomy background, or like here because you're like interested in processing like astronomy pictures? Originally, or like I'm like a biophysicist, something. I'm in the chemistry and biochemistry department, but I don't know anything about chemistry or biochemistry. I'm originally a physicist, and I work mostly on biology. So yes, this is me. So no, not much astronomy. Oh, astronomy, there you go. How many of you are like biology, microscopy? Almost everyone, yes, so that's what I assumed. What else? What am I missing? Medicine. Okay, but like what types of images? So mostly like these kind of images?
Yeah.
Okay. So like biomedical imaging.
Yes.
Okay, it's the same approaches would work for all of the above. And so you see there is, even in this kind of small group, it's somewhat diverse. And is anyone from, I'm assuming not, but is anyone like from computer science and does like natural like text editing and stuff like that? Or like text recognition? So that's also something that people might do, like, you know, go over medical records and try and extract information from that. All right, so diverse stuff you can do with image processing. So let's very briefly talk about kind of the formats of images that we often work with in science. So largely speaking, images that we save on our computer can be either lossless, meaning we save all the information on a single pixel level for every single image, or lossy, where we find some ways to compress this data. But of course, that leads us to lose certain amounts of the data. So if you ever heard of like JPEG, so JPEG is actually a compression algorithm. So JPEG images are lossy, but if you heard of like PNGs are not lossy. So they save image data on a single pixel level. Many microscope makers have their own proprietary image types, but for the most part, at this point, we have open source ways of opening more or less any image type. These are some common ones. So I think JPEGs we often don't use in science, aside from, you know, to make sometimes presentations if we want to make them smaller. Pings are somewhat common. TIFFs are very common, and then you have all these different proprietary ones. Specifically, one that is becoming more and more important in the last few years are these HDF5 or N5 or any of the kind of hierarchical data formats, which are actually designed to store very large data sets. So as soon as your data sets become on the order of many gigabytes or even terabytes, saving them as keeping them as individual TIFFs becomes kind of impossible, because just run out of space. And so people came up with these hierarchical data structures that are lossless. So we still can maintain all the information, but we do it in a kind of more clever way that saves everything in a kind of a pyramid scheme, so to speak, where different resolutions are saved at different levels, and so that kind of maintains smaller data sets. Okay, so what tools are we going to be using here? We're going to be using, I should have put Google up front, because this is the main tool that people use for programming anything. So we're going to use our computers and Google. We're going to use Stack Overflow to look for solutions for problems that we're going to encounter. We're going to be using Python to program. And then in terms of specific packages, yeah.
Sorry, what software do you use to convert like images to these hierarchical data formats?
So there are specific software that the HDF5 group is putting out to convert this.
Yes.
But at this point, there are packages for MATLAB and for Python that allow you to do that. So if you want to talk about it, we can talk, like, because this is niche a little bit, so we can talk about this later. This is actually something that I kind of have to do a lot in my stuff. So also maybe I should have given some background on myself. A lot of what I do is imaging. I build and operate microscopes. And so the microscopes we have, it's actually right here in the back, are light sheet microscopes and regular epifluorescent microscopes. The light sheet microscopes generate data on order of multiple terabytes per experiment. And so having compression, lossless compression for these data sets is very, very important, because our main bottleneck is data storage, strangely enough, which is the thing that most people don't even think about, just buy a hard drive. But we don't have money to buy 100 hard drives. So, so if you want to talk about this later, we certainly can. We're going to be using scikit image, so this image processing toolkit for Python. We're going to be using Jupyter. This is where we're going to be doing all of our work. We're going to be using NumPy, which is a matrix. I mean, I don't know why I'm saying this. I'm going to say this again in a minute, so let's just go there. One of Python's major advantages is it's free, and also that it has this very extensive set of libraries that can do more or less anything. And these are what we're going to be using. So NumPy is kind of a very advanced matrix calculator. It was actually motivated by MATLAB, if you guys know MATLAB. So many of the functionality is similar, but the idea is to have an efficient way to work with matrices. So matrix multiplication and vectorizing is kind of the core. We're going to be working with matplotlib, which is a graphing, plotting, and viewing package. So this is how we're going to be presenting all of our results, right? We're going to be working with scikit image, which is this high-level image processing. So when I say high level, do you know what I mean? When computer scientists say high level, does that mean it's very complex? Or does that mean it's very simple? Simple. So high level means that all the hard work has been done for you. You need to implement very little algorithms on your own, and there are a lot of tools that are already built in the way that you just call a function that somebody made. And Python itself is a high-level programming language, so we are not dealing with moving bits around. We're dealing with calling functions, right? So, and then we're gonna very briefly touch on pandas, which is this data management package. And you can imagine that as soon as we convert things from the level of an image to the level of data and basically tables, then we're going to need something specific to deal with this type of data. And pandas is kind of the tool of choice for these kind of things. Now, kind of the structure of what we are going to be doing over the next 3 days. So we're going to go over the setup and downloading everything and making sure we have we can start doing things in a few minutes. But when we download the course material, I'm going to find 2 notebooks. One is going to be called the teacher notebook, and one is going to be called the student notebook. The teacher notebook is fully completed, so you can go over it and see everything in it. And if you have really no experience at all programming, and you're kind of really scared of trying, you can do that. This is less recommended. The student notebook has a lot of open parts in it, so stuff that you will need to fill in. You're strongly encouraged to work in pairs or groups. So you actually, it's been scientifically proven that you learn more working in a group than you do working alone. Use Google as much as you can. Also in the notebooks, there are links to relevant manual pages for specific functions that you're going to use. So this is a workshop, meaning you're going to be working and I'm going to be working. This is something that we do together. in the workshops, in the notebooks, there are already links, but also if you're not sure about how to do a certain thing, Google it. This is how people work in the real world, right? You don't always have someone to ask or a textbook. Google is your friend. It's also good if you work in pairs, for instance, one person to be doing the coding and one person to be doing the Googling, and then there is some discussion, and then you can switch back and forth. So this is actually a method that's used in industry for programming, because this is how you can find bugs, and you can find, it actually makes the work more efficient. So it's kind of surprising, and it's maybe good to learn how to work like that. But you're free to do whatever you want. So if you want to work alone, that's good. If you want to work in a group, that's good. Your call. We'll be going over everything together, but we're going to leave time between sections for you to try and implement things on your own, to do things on your own. I'm going to be going around trying to help if you need help, or just discuss things if you just want to talk. And at the end of every one of these blocks, we're going to go over things and do it together on here. Okay, so does this make sense? Okay, okay, let's get going. So we need to download or clone the course material. So I sent you a link yesterday, but even if you didn't get it or you didn't download it, that's fine. By show of hands, who here got like Anaconda going yesterday or the last few days? So any, like the email I sent out yesterday, who didn't? Maybe this would be easier. Okay, that's fine. I mean, if you have all the packages installed, that's fine. And if you have Python already, and if you have like Conda or pip on your computer, that's also going to be pretty easy to set up. But okay. So we're going to just be using the Jupyter, not Jupyter Lab, just Jupyter. So if you have Jupyter, that's good. If you have Jupyter Lab, you probably also have Jupyter. Yeah, so Anaconda has Jupyter in it. So if you download Anaconda, you're definitely good. If not, then you're probably good. And if you're not, then we're going to solve that. Okay, figure it out. You can also probably work on that computer, and that computer definitely would have Anaconda. Okay, so the only thing you probably didn't do yet, maybe you did, maybe you didn't, is to actually get the course material. So please travel to this place on the interwebs. So I sent you this link yesterday. You can just go to the email you got yesterday, and if not, you can just type this in. Okay. Maybe it would be easier. Did anyone here not download that yet? You didn't. Okay. Cool. So let's go over this. So anyone who needs to get this, are you on this page right now? Okay, so let's wait. It's good. Thank you. So is that computer working? And it has Anaconda on it? So you probably find it under like applications, and then it's going to have the like Anaconda navigator or whatever it's called. Okay, so once you make it into this page, please click on clone or download. Okay. Then say download zip. Okay. Save that zip somewhere, like documents is a good place to save these things. You can save it wherever you want. So take it there, then unzip it. And you should have a folder with all of the relevant coursework on your computer. Does everyone have a folder with all the relevant coursework on their computer?
So can we put this folder anywhere?
Anywhere. Anywhere. But remember where it is, because you're going to need to get there eventually. You're gonna
Anaconda is launching.
launching. Okay, cool. So it's there. So it should be okay. You can download the coursework. So the course material. Again, this is where it is. So just open a browser, go to github.com/Alonian, which is me, /DIP, which is digital image processing, and just download and save it wherever you want. Okay, and if we have that on our computer, then we should have everything ready. So let's get started with Jupyter Notebooks. Okay, so go to, okay. What are Jupyter Notebooks? Jupyter Notebooks are these special documents. They're called a live code document. They allow you to kind of create and share code relatively easily. They're a really good way to keep your kind of coding projects somewhat organized. It's not good for everything. So I actually have, I think Jupyter Notebooks are for the most part overused because people use them for these like massive infrastructure projects that just become unusable because it's not very modular and people aren't implementing things in kind of a good computer science way. So the structure of the code base is not good, but it's good for trying things out, and it's good for kind of experimenting and prototyping and stuff like that. And this is mostly what we're going to be using it for today. So we want to start Anaconda. So go to either search for Anaconda Navigator or go to Applications Anaconda Navigator or wherever that is. Once you open Anaconda Navigator, click on Jupyter. So this one, not Jupyter Lab, Jupyter. Right? Alternatively, you can just open a terminal and type Jupyter Notebook, and that would also open a Jupyter Notebook. Once you do that, your browser is going to open a window that looks kind of like this. Is everyone more or less there? Is anyone not there? Has anyone like really lost me a while ago? Did anyone lost me a while ago and not sure what I'm doing and why? Okay. I'm never sure if the silence is because I'm like completely gone or is it like because, so give me something. You're good? You're good. You're good? I'm gonna say, you're good. Good? Good. Okay, cool. Yeah, of course. Yeah, this is my computer. So it has files and your computer has different files. Okay, so the file browser is just like our computer. And just navigate to wherever you put the repository, okay? So you can just navigate normally the same way you would navigate on your own computer and get to wherever you put your repository. So I put it in Documents, Repos, BIP, Date, Jupyter Notebooks. So get to this position. Again, on your computer, this is gonna be wherever you just saved your repository that you got from GitHub. Does anyone here use GitHub on a day-to-day basis? Just so I know. Yeah, good. Anyone who doesn't, you should probably think about it because it's a very useful tool that makes life very easy. To share code and keep yourself updated. So a repository is a directory, a folder with a bunch of code programs that are related to a certain project. And there are certain software. So the one you just downloaded, the zip file you just got. So dip, master, that's a repository. So it's just a bunch of code that's sitting on some server on the internet. Okay, and okay, so everybody's here. We got these two notebooks.
Should be a special folder? I don't know, because I put it in one folder that I
wherever you put it, this is where you should find it. Did you unzip it?
Yeah, exactly. But I cannot find it through the
Okay, so did you get it? Can you? Okay, cool. Okay, so now is decision time. Okay, so you get to decide whether you want to be a teacher or a student today. I recommend that you be a student, but if you choose to be a teacher, that's also fine. Okay. And again, it would be very useful for you to work in groups or in pairs. You don't have to, but you can make that decision in a few minutes as soon as we start going. You can also mix things up. You can work in groups on some parts and alone on other parts, and because we're going to have a lot of blocks of individual work coming up. And at this point, we're more or less ready to start processing. So trust the process. This is very important. So let's go. So I'm going to go and do the same thing that you just did. So I'm going to go to here. I'm going to open both, if you don't mind. So does everyone have something that looks like this open on their cool. So we're good to start. What time is it? 9:42. So do you guys want to take like a 5-minute break before we actually start things? So let's take a 5-minute break just to make sure that. Oh, you have a question? Sorry. Again, sorry. Okay, so this is a complicated explanation. So what you're actually doing here when you're opening a Jupyter notebook is you're basically opening a server. And that server is some address on your computer. So you have ports on your computer that you can connect to, and one of them is port 8888. Localhost is your computer, and you're connected to port 8888 on your localhost computer. And now you can actually access this from anywhere, effectively. So one of the nice things about Jupyter Notebooks is that you can, once you open that server, you can connect to that server from many different places. And the easiest way that or the most reasonable way that humanity came up with to connect to locations in the world, other computers, other servers, is through kind of the internet, right? And so effectively, you're connecting through the internet to your own computer, to a server on your own computer. So that's why it's all on the browser. What? Yeah. So let's take a 5-minute break. Make sure everybody is up to date. I need to go get.
Yeah.
So these iCard notebook files, you can only open it using notebook. So without it, it's clicking on some work. So do you have Jupyter notebook on your computer? No. Python? these are still open computers here. So there are still open machines here I can use. And then if yeah, you can use this one, and then this one definitely would have it installed already. And then I'm going to ask someone to help you download the repository. Okay. The address here. More slides. What?
Talking about your presentation, your PowerPoint.
No PowerPoint. We're now doing this. When I do this, do you see it?
It should be fine. You should see it. It's like it shares pause. So you go here, you click that. Where's that program you're using? And then you click share. And this should not be. Okay. Is this what you're trying to share or no?
Yep.
Jupyter?
Yeah.
Yeah. As long as this is green, you'll be able to see that.
Cool. What up?
So you want to share this, right?
Yeah.
Okay. Well, this is green. Okay. Yeah, you're good. So now Carolina, see that.
Okay. So we're going to start with a little bit of a warm-up. Now notice that there is a little bit of a disclaimer here. So when we're working with Jupyter notebooks or in Python generally, we always have to load whatever software package that we want to use. It's good practice to load these packages once and only once. So if I'm going to use NumPy, I'm going to load it wherever, like at the beginning, and then use it throughout. However, in this case, I keep reloading it in multiple places, basically every time I use it. And this is just to make sure that we can kind of jump into any point and start working, and we don't have to run everything up to that point before we can start, before we can go on. In general, that's not really good practice because it just adds a lot of overhead. So every time I load a library, it takes some time, and that time is time that I won't ever get back. And I'm just gonna, so just keep that in mind. And the very first thing we're going to do is kind of a warm-up. So we talked about what exactly images are and how computers store images in their memory, and we kind of want to see exactly, get a sense of what pixels are and what images are from the point of view of computer. So the very first thing we're going to do is we're going to try and make an image of just a checkerboard. Right? So checkerboard, you all know what it is. And now if you look at the code, so this is a student notebook, and you see that there are a lot of places that are kind of empty. So if I try to run this, it's going to not run, right? And it's your job to fill out the parts that are not running. Right? Oh, sorry. So to run a cell, so each one of these blocks is called a cell, and to run it, you go Shift-Enter. Right? So if you put your cursor in this block and just go Shift-Enter, it's going to try and run it. And if it fails, it's going to tell you, oh, I failed. In this case, it's failed because there is an open statement somewhere here. Okay? So is everybody getting this? So if you just go Shift and Enter, you fail to run this cell. Shift and Enter together. So put your cursor in this block of code and just go Shift Enter.
Is it the same in Max? It's not working.
So shift, yeah, it's the same.
Okay, at the same time.
It's the same time. Yeah. Shift and enter at the same time. Just click them together. It's usually better to do shift and then add the enter, because if you try to enter and then add the shift, then things just move around. So okay, so we're going to take the next 10 minutes to go from 0 point nothing here to 1 right here. Over this, what you're going to need to fill is here. So this checkerboard thing, right? And at the very end, you're going to need to present a certain image right here. So everything else should run smoothly, but these two things you need to try and fill yourself and make it run. So let's take 10 minutes to go from 0 point nothing to 1. So this is kind of a warm-up, kind of to get you familiar with what pixels are and how we play with. Okay, so let's do that. Set a timer. Yep. With code. Use Google, and in 10 minutes, I'm going to tell you how. You can always look at the answers in the teacher one if you really want to. But so this is how it works, right? This is how in reality things are gonna happen, right? You're gonna get something that you need to do. You're gonna start off by not knowing how to do it. You're gonna try and teach yourself how to do it. And it's okay. So look it up. It's like you can also work in pairs, or if you really feel like you don't even know where to start, you can try and get hints from the other notebook, right? So the other notebook already has everything filled out. I'm going to give some hints as we go along, okay? So
I have a question. Is there like a document or something that tells us what functions are in NumPy?
NumPy has like what's called an API. So there is a document that tells you all the functions in NumPy. You can just search NumPy API and see. So first hint, a dark pixel is going to be 0, and a bright pixel or a white pixel is going to be 1. So you're going to have a grid of mostly of zeros and ones, right? Because checkerboard is 0 and 1. And notice that I said this is going to need to be a 9 by 9 grid, right? There's going to be another hint in 2 minutes. Let's start. You can add more lines, by the way. You don't have to stick to one line.
So the purpose is how to make matrix.
So you need to make a 9 by 9 matrix with zeros and ones in the right places.
How do we know where to put the ones?
So you can choose either having ones like either start with like bright pixel at the corner or dark pixel at the corner, or just like I don't care about the orientation of the. Yeah, it doesn't. This is again just for kind of practice and understanding what pixel does.
So functions is
NP dot array might work. In about a minute, I'm gonna give like another clue. And so when I write import numpy as NP, that means that I just imported everything in NumPy and called it NP. So now if I want to call a function out of NP, I call NP dot whatever the function name
Did you
is.
say numpy dot or it's still work?
I don't think so.
You have to use it.
So we already did, right? So we've imported NumPy here.
Right.
So I call the import NumPy as NP.
or you just import numpy somehow.
So after you import, you can use it any function from NP by calling NP dot something. And this is our next clue. The function NP dot zeros creates a matrix or an array, let's be technical, of zeros. So this is a good place to start. You start with an array of zeros, you just need to fill out which ones are not zero. Right? So this is a good place to start using the function np.zeros. Did anyone already get it to work? Can you show me? I'm going to come there. Yeah, you got it. So remember, we're going to go from 0 all the way down to 1. All right, so there's one more thing to fill out. You're going to want to just present this image that we loaded. So this should run smoothly and this should run smoothly. And then after that, you're going to need to just plot this image that we loaded. So you should already have an example on how to plot something in the lines above, and then you just need to kind of modify it to your needs. So the ones whoever finished already, ask around if there are people around you who might want some assistance. This can possibly be good pairings if you are interested in working in pairs. So so we're going to go for it. But you know what?
Yes.
This is how coding is. And so what you can do is did it run all the way to the end?
No.
Oh, so if you finish in about a few minutes, you can try and figure out what exactly you did there. What exactly did this action? Are we good? Are we stuck? All you want to do is you want an output. So you want an output of this function being x. So right now this function is spreads. You want to go to the output of your ring, so you can say or return x. Return x. So this should return x, and then you can run the function to have an output. So when you do this, so normally x equals square root of x. Okay, so all right, so let's go over this thing. Okay, so like I said, we're going to start by making an array of zeros. So this is just by saying np dot zeros, and the array is going to be 9 by 9. Cool. So this is just going to give me a bunch of zeros. And then I want to put ones in the correct positions, right? So the correct positions are, I start with 0, 0, and do jumps of 2 in each direction, right? Or I start with, 0, 1, and 1, 0, and do jumps of 2 in each direction, right? So if this is my array, and finally I want this. So I'm going to start at 0, 1. So remember that in Python, all the indexing starts at 0. So I'm going to start at 0, 1. So let's see here. And then do jumps of 2 in each direction. And each direction here, and each direction here. And I'm going to do the same with 1, 0, and do jumps of 2 in each direction here, and each direction here. And for all these combinations. So the way this is done is to say that checkerboard. So someone asked me about indexing. So the way indexing works is I said I'm gonna start at 0 and at 1, right? And I'm gonna go all the way to the end. And jumps of 2. So the way the syntax is where you start, colon, where you end, colon, how many jumps. So what jump in every step. So I'm going to start at 0 and do jumps of 2. I'm going to start at 1 and do jumps of 2. So this is going to give me all the columns. And I'm going to do the same for starting at 1, 0. And all of this is going to need to be 1, right? So I'm putting 1s at all the positions that are, I did it the opposite here, but like here, 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 throughout the whole matrix. Or not. Zeros, I just didn't call zeros right. What did I do? Oh, yeah, sorry. Yeah, the size should be in parentheses. Thank you for the autocorrect. So you see I get this matrix of 1s and 0s. All right. Now I can take this matrix and show it. And you see how a matrix of ones and zeros corresponds to an image. Did you stopped at the matrix level? Yeah? That's okay. So this part, if you got the previous part right, this should just run normally. So this ones and zeros corresponds to an image, and that image is this checkerboard. All right. Now we're going to do another little thing like that. We're going to load one of these test images. So scikit image. This is how we load it, and we import just one part of it. The part we import is called data, and this is just test data that you can use for whatever you want. This should run smoothly, like you don't need to change anything. And we're just going to print all the numbers, right? So any image that we load is just a bunch of numbers, right? So we want to convert this list or array of numbers into an actual image. So this is should be an image of a bush. And we have this part where we are supposed to show it, right? And so the way we show it is kind of similar to how we showed an image beforehand. So if you go up, you saw that I use this function imshow, right? So I'm going to just copy it. I'm going to put it down here. And instead of the checkerboard, I'm going to put the image of a bush. Did everybody get an image of a bush? And to show the image, I actually need to call the function show. So once I run this, I get an image of a bush. Okay?
I think I have a fundamental problem that my modules aren't being found.
Your modules aren't being found. Which modules? So I put a color map gray. You can put whatever color map. We'll figure it out in the next
Really?
problem. Yeah.
I get attribute error when I run the code. I don't get a matrix of numbers.
In which one?
The previous one, the cell before this.
This?
Yeah, I'm not able to run it. It gives me an attribute error.
Where?
Yeah, that part like there's no attribute in show. Did you just work?
Here?
It's a UK.
The one after the checkerboard. Yeah, this one.
So this one, when you run it, so there is no imshow here, right? Oh, so here it doesn't load the image. What did it say?
It gives me an attribute error.
Attribute error. Okay, so this is why we're doing these warm-ups, because this is when all these little things pop up. Okay, and so we're gonna figure out all these things in a minute when we go over the next step. We're gonna make sure everybody is in sync. Okay, so okay, so the simplest, so don't worry for a moment. We're going to figure it out in a minute, okay? And during the next kind of work session, we're going to go over and make sure everything is right. All right. So the simplest thing we can do with images. We good? Yes, no? Yeah.
Is the image of bush like stored in that file stored somewhere?
Again?
Like is it stored in those folders that we downloaded?
So this specific image is, so this image processing thing, scikit image, has a bunch of test images built into the package. So it's stored with the package. These are really small images. They're like 25 by 25 or something like that. So they don't take up a lot of space. We are going to use data that we have in the repository that we downloaded.
And then like this image, we have that file in our directory.
So
Where that come from?
So if you downloaded an Anaconda, or you have here, so Skimage is scikit image, so it's an image processing toolbox basically for Python. And if you installed Anaconda, it's pre-installed there, but it's a package that you call. So you have a list of packages on your. This is actually something we can do. If you go to any terminal window or a command line and type conda list, in the terminal window, this would list all of the packages that you have installed with your Anaconda. So if you just curious about all the different things you can use, this would give you that list of all the different things you can use. And you can always add to that list by installing more things. So, but we're not going to go too deeply into package management in Anaconda. We're just going to use mostly things that are already built in. So this data set actually has many different images, and we're only showing one of them. So we're only keeping one of them. So I'm keeping the 0th image. So the first image in that data set actually has like 100 images of other stuff. You can put any number and it will plot just different things, different faces, mostly. All right, so the next thing we're going to do is pixel level operations. Let's go on. Let's go. Oh, yeah. Let's go on and we'll fix that during the next session. Okay. So pixel level operations are kind of the simplest thing you can do with an image, right? So all it is like I'm going to take individual pixels and apply something to these pixels. I can invert them, meaning turn ones to zeros and vice versa, or 0.2 to 0.8, so the opposite, 1 minus what I have. I can apply some function on them. I can square, I can anything I want. And it's kind of the very most basic thing you can do with an image, right? You're only going to an individual pixel. You don't care at all about the environment. You're just doing something to that pixel. And this is what we're going to start off with. We're going to look at a slightly more interesting image than that image of a bush. So that same package, that same module and scikit image also has some immunohistochemistry images. Okay, so this should run with no problem, right? So if you go over this, I'm going to open this image and it's going to show me this image, which is some of you might recognize as just an immunohistochemistry standard image, right? So everybody has this image on their screen, or everybody who has things working. We're going to fix that in a minute. Okay, so first of all, we can already see that this is a color image, right? This is RGB. We can look at every one of these channels independently. So again, this is just kind of should run. Oh, sorry, it shouldn't. So we're gonna, I guess, implement this in a minute. So let's look at each one of these channels independently. So if I look at channel everything, and 0, this is the 0th channel, right? So take a minute and fill out the rest of them. And at the end, you should have shown 3 images of different colors, red, green, and blue. So this is the first one, and continue filling the rest of them, okay? So what's that?
I've got an error message on this image now. So I tried to put that in, but then I got a message suggesting I put this still line instead. So that's what I'm trying to do at the moment, but it's all up.
Okay, so you mind if I
Sure.
close your... Anaconda. Image, you have second image. It should be here. This is how you started, and you start from somewhere else. Like,
I just clicked on.
oh, okay. Let's open the teacher one.
Awesome.
Okay. So now if you open the student one, okay.
Thank you.
Okay. So I'm going to get to, did you figure it out or no? Okay, so I'm going to get to you in a minute. So
Yeah.
does everyone have 3 different images on their computer of 3 different channels? Yay, nay? Did you get something that looks more or less like this? Yes? Yes, cool. So for now, we're not going to worry too much about RGB. Okay, so let's stick to one color for now. The next cell here, all it does is just picks out the first channel. So we're only going to work with that. And this is the channel we're going to be looking at for the next few minutes. Okay. And in the next 15 minutes, I will ask you to go from where we are right now, right, until 1.2. So until we get to a section called image filtering. So I'm going to put a timer for 15 minutes. And at the end of these 15 minutes, we're going to try and solve this ourselves. And wherever it doesn't run, you should try and fill it up. For instance, here you need to invert an image. And I already gave you a hint that there is this function called invert, right? Now notice that there are links everywhere. All right, so you can follow these links and see some functions and how they work and what their expected inputs are. Okay, so in 15 minutes, 1.2. Okay, I'm going to come and try and help you out. Possible the version of the person who installed Congress computer installed their lightweight version of it. Doesn't have all the built-in stuff. So I'm just adding all the features. Which sometimes is, there's no function of 3 gigabytes of stuff. Doesn't mean Let's keep it to So how's it going, everyone? I'm glad to announce that everybody has everything running. That's good. Does everybody have already an inverted image on their screen? Yay, nay? Yes? Inverted, yeah. Other stuff, other transforms? Now, if you finish ahead of time and you have some time to spare, you can look at other stuff from the exposure module and see if there are any interesting things that you can play around with. And just feel free to add whatever you want to add. It's your code. We also see how using these high-level libraries can make our life very simple because we don't need to know what an exponent is or how log functions work. Although you can also try, if you want to implement these things yourself. So instead of calling log transform, to just call numpy.log, for instance, or whatever else you want to do.
Sure. What I'm confused about is trying to figure out, okay, so when here, because I just camera, but I just looked in the teacher one, it says exposure documents. So when do we have to use the sensor stuff, when do we not?
So structure is you have a package. This package is cited. Every package has a set of modules. So exposure is one of the exposure, the filter, the data.
And each module has function.
And each module has function. When you call a module, you want to call a function out of that module, you have to specify if you're calling the function out of that module. The exposure doc is function. You could have said from scikit.exposure import just now. So you can say from that specific module import this specific function. So this is what we did above with invert.
That's why you don't need it.
And then you don't need it.
Okay.
So this is the structure. So we said from the detail import this now.
Yes, thank you. And then just another question is, so okay, so this is a package.
This is a package.
This is the numpy is a
Package.
that's also a package.
Yeah.
So why do you say from here and say import here?
So from here, I'm importing only certain module. I'm only importing the data module from site.
Okay.
Here I'm importing the whole package, and I'm just calling it up. So you can also change the name of how you import things. So you can say from scikit import data as something.
Okay.
But so I don't have to rewrite all this every time. I can say call this whole thing guild.
So here you go, so like import data
As guild.
Okay.
Is anybody working in pairs? I'm just interested to know. You guys? Small groups? Groups of one? So, So fun fact about group learning, people who learn in a group demonstrably do better, even though they think they're learning less, just so you know. That's true. So when you're in a group, you think I'm not learning as much because other people are around me, and you end up actually learning more than you would individually. So just take that into account. So did you go here? I remind you that both Google is your friend and helpful links I put in there are your friends. So if you're not sure on how to do something, looking at these links would probably help. So
yeah, because I changed the number and then say
so it doesn't define how dark it should get, but it defines how much you. So it's not a linear. So it takes the input to some power. If you have numbers that are smaller than 1, so I mean any number, the higher the power, the more it's going to stretch the difference between the brightest images and the darker images.
so it is a contrast.
So yeah, it changes the contrast in that way. So let's say gamma is 3. You have 1 pixel that's 1, another pixel is 0.5. So before you did anything, the difference between them was 0.5. After you did a gamma transform with gamma equals 3, 1 stayed 1. 1 to the third is 1. 0.5 to the third is 1/8. Right, 1 over 2 to the third is 1/8. Now the difference between them is 7/8. So you took a smaller difference and you made it a bigger difference. So the bigger the gamma, the more you're going to stretch it out. If gamma is smaller than 1, you're going to squeeze it.
the squeeze of difference.
So if gamma is smaller than 1, then the difference is going to be smaller than what you started. So, so if gamma is 0.5, this is a square root, right? So square root makes large differences smaller. So that's the idea. Okay, so let's go over this. So there was a very good question about exactly the structure of packages, modules, functions, right? What exactly are we doing when we're saying from this, import this, so on and so forth. So the structure is so we have functions. Functions do a specific thing. They do a specific thing. Okay, let's say inverting an image or applying some transformation, whatever the image is doing. If you take a bunch of these functions and collect them into a group of functions sitting in one big file, this is called a module. Usually you're going to try to keep all of your modules, or like each module containing functions that do more or less similar things, or like things of the same nature. For instance, I'm not going to have a module that does plotting and fitting, right? That doesn't make sense. I'm going to have one module for plotting and one module for fitting. Okay, but if I want to design a whole package, so a package is going to be a set of these modules. If I want to design a package for scientific programming, I'm going to need to have both something that does fitting and something that does plotting, right? So the way to keep everything organized is to have the package, so it's named, it packages different modules together for kind of a common goal. So the hierarchy is that package contains modules, modules contain functions. Now when we say import, let's say the package NumPy, so this is going to import everything in NumPy, right? Now I can import it and give it a different name. I can say, let's say I don't want to write NumPy every time I call something from NumPy. I can say, after you import it, call every function, call that package NP. And this is common. Now if I want to call a specific module from NP, from NumPy, I'm going to have to say NP dot whatever the module name is. And then I probably want to have some specific function from this, so I'm going to need to do dot whatever function name is. You can imagine that this can get a bit laborious if I have to call the same thing 700 times, and every time I need to do all of this. Instead of this, I can say I only need a certain module out of NumPy. I don't need the whole thing. So I can import only that specific module. So let's say from NumPy, I want to import a module called something, banana. So I say from NumPy, import banana. Now I don't need to write all of this part. I can just call specific functions from this module banana by calling dot function. Now I can also import banana as, give it a different name. I can import it as bn, and then instead of banana dot whatever, I can call it just bn dot whatever. So if I'm going to use multiple functions from the same module, but only from that module, I'm going to import just that module. Now let's say I don't care at all about the module and I want one specific function from one specific module from one specific package. So I do, instead of saying from here import banana and have the whole module, I can do what I did here. So from scikit.util, so this is, what is this? This object, scikit.util, it's a module. So from this module, I'm going to import a specific function. that function is the function invert. All right. Now I can just use the function invert as is. I don't need to tell the I don't need to tell the computer which invert to use. I'm going to use this specific invert because I already imported it with the name invert. So let's go over questions about this. Yeah.
You also have import matplot live.pyplot
Yeah.
as guilty.
Yeah.
So is the matplot, whatever, matplot live.pyplot, is that a module?
Yeah.
Okay, so it's similar to that.
Yeah. So I can say import. So instead of this, I can say this is equivalent, I think, dot banana as bn. I think these are equivalent things.
And is a library the same as a package?
Yeah.
So if you call package, there's a module is from.
So if I want to import a certain part of a package, I need to use from, because from that package, I'm going to take something. If I import the whole thing, I use import and usually give it a name. Okay, so the first thing we wanted to do is to invert this image. How should I do that? Ideas? So I just use the function invert that I just imported. And this is the image I want to invert. And this is the original, and this is the inverted image. Did everyone get something that looks like this? More or less? Okay. Now, we went and instead of just kind of a simple inversion, we wanted to apply a bunch of different pixel level operations. So we use this exposure module. Did everyone go click the link? I worked very hard to make sure there are links everywhere. And from that link, you'd be able to see that there are all these different interesting functions in here. And specifically, you could just pick out the functions that were asked for over here. Now notice that right now, I didn't import each one of these functions individually. I imported just the module exposure. So exposure has all these functions underneath it. So I said I want gamma equal 2. And then exposure dot something like that, right? Gamma corrected? No, just gamma. Just gamma of image hist and gamma. So if you looked at the manual, basically, you saw that the function adjust gamma has two inputs. One is the image, and one is which gamma do you want to use. So which what number to put here in the whatever. Okay, same goes for the rest of them. So exposure dot adjust log. This one doesn't need any other inputs. Here I said I want a cutoff of 0.4, and I want a sigmoid scaling. So sigmoid looks kind of like, like an S shape. So exposure dot adjust sigmoid of image hist. And I specifically said I wanted a cutoff. So I'm going to say that the cutoff is the cutoff. Right? So far, so good.
Just a quick question. So in the gamma one, you don't specify what the value of the gamma is, but for the cutoff you do.
So gamma forces you to have two inputs. Here, I don't think you have to have two inputs. I think this is like, what do you call it? Like an optional input. And to put optional inputs in, you have to put them like that. I might be wrong. So it's possible I can just give the cutoff. But you know what? Let's check. Exposure, adjust sigmoid. yeah, so you see that the cutoff here is an optional thing. Otherwise, it just takes it as this 1 or something. Oh no, 0.5 versus just gamma, which is somewhere around here. Yeah, you can probably do the same thing.
Yeah, I just tried it.
Yeah, you can do the same. Sure. I guess it knows that the second one, if you don't do it like that, is gamma. That works equally well in this case. Probably this is a bit easier to read your code. So if you do it like this is probably a better way of doing it. The next thing we want to do is equalize. So equalize basically maps the histogram of your pixel values into a constant or like a uniform distribution. Usually what it does is it stretches out your constant contrast. So it makes your darker things darkest pixels darker than they are, and your brightest pixels brighter than they are, right? Because it takes the brightest one and puts it at 1, no matter what it is, and the darkest one, it puts it at 0, and everything in between just flattens out. So it has the effect of increasing contrast. This is so exposure dot equalize hist of image hist. Okay, and this is a kind of more let's say advanced one, which it kind of does the same thing that equalize does, but equalize has the problem of if you have an image that is has the very different brightness on different regions of it, then it treats everything as one thing, and then like if you have, it would make half of your image dark and half of your image bright, for instance. If the if the bright pixels are on one half and the dark ones are on the other. So this is kind of a slightly modified algorithm which applies equalization of the histogram locally. So for every region, it kind of does its own localized equalization. So this is exposure dot so adaptive equalization it's called, and image hist. So everybody had more or less this going on? Yes, no? Was there any specific one that anyone had issues with, or something that didn't work, or the syntax was unclear? So let's just see that this works. Of course it won't. Oh, see. So you see these like simple corrections, and you see, for instance, how equalizing does what we thought it would do. It takes the darkest pixels and makes them darker, the lightest pixels and makes them lighter. You can play around with the cutoff for sigmoid and see how exactly it affects your image. So sigmoid basically also is a way to increase contrast by saying, I'm going to take everything above a certain level and effectively make it almost 1, and everything below a certain level and effectively make it almost 0. So you can see this, how this image is really, really darks and really, really brights, and only there is like a thin range around 0.4 of the original intensity levels where this is not the case. Now you also can see the effect of this localized equalization. You see how here, because all these pixels are in the same place and they're all dark, this kind of looks just like a black blob here, and you can't really see any details. But if we just locally adjust this contrast, you see that the details are preserved, but the overall contrast of the image is increased, right? So this is pretty cool, and these adaptive algorithms are often useful for many real-life situations. So questions so far? Yeah.
Your experience applying these different contrasts, they make a huge difference in data analysis.
It kind of depends on what exactly you're trying to get. So, and this would be more or less my answer to almost every question in this class. So it's very different, for instance, if all you want to say is like, what of this image, what are the dense cellular regions? If you only needed that, then clearly this gives you the highest contrast between dense cellular regions and less than cellular regions, right? So you do this, you say, okay, I'm going to find continuous regions above a certain size, and I'm going to say these are mine. If you really care about identifying each one of these specific cells here, then maybe this is actually good because you took away everything else. Maybe I want to invert it before doing it, and that would give me. So there is a little bit of like trial and error in most of these algorithms. Right. So let's again take a few minutes to refresh ourselves, and we'll come back and talk about what is probably the most heavily used tool in image analysis, which is image filtering. We're going to only give kind of an introduction to it, and I'll give a few examples of things that go beyond the scope of this class, but at least I will talk about a few of these examples. But this is kind of the core of image analysis is like a lot of image filtering. So let's take, questions?
Looking at the documentation they had online, they had a lot of different properties or options that you had while going through and using these equalization or adjustments.
Mm-hmm.
But how did you know if it was a required input versus a suggested input?
So I'm pretty sure none of the ones I used here were required because I think there is a default gamma. But if something is required and you run without it, it would not run. It would just tell you missing this many inputs. So they will let you know.
Just wondering, would it be possible to have a bunch of these different exposure filters layered onto one image?
Again.
Can you do adjust the gamma and then the cutoff?
Of course, yeah. So you can do this, and then you can take this gamma corrected image and feed it into the next one. And most image analysis pipelines actually work like this. So you do one thing, and then you take the output of that one thing and feed it to the next thing, and so on and so forth. And it kind of is built like kind of a nesting doll.
Yeah, I tried doing that, but I think I got the syntax wrong.
Let's see. Let's just take this. No worries. This is good. Copy. And just let's put this here. So now I took this gamma corrected image and fed it to a log transform. So what is here now is the log transform of the gamma corrected image. Right?
Thanks. Sorry.
No worries. No need to apologize ever. Cool. So you can take the output. So if it's an image, you take the image, you feed it to something else, and you get another image. You can do as many of these as you want in whatever order you want. Cool. All right. So let's take a few minutes. Like 7 to 10. So if you don't want to break, we don't have to. Like if you want to just keep going, we can do that too. If you do want a few minutes, we can do that. We want to break. Okay, cool. You're all sitting, so I was like, okay, maybe they're like having fun.
Yeah, checking the mobile.
See what's up on the Twitter. Oh, so the same size is one. The size for 0 is 1. This is
It was really cool. And that's the constant use But like when I looked at courses, they broke it up into sections. Like the first half of it was
Very good. Very good. Much more good.
Okay.
But that one, they didn't really force us to actually do very much. It was like, it was sort of like this, like most of it started like zero and
Did everyone sign the attendance sheet?
Yeah.
Not signed the attendance sheet. Something
Everybody
very bad
signed
is
this.
gonna happen.
Everybody signed this guy over there.
Something very, very bad. Terrible things will happen if you don't sign the attendance sheet.
But the nice thing about like the college and Monday Mud is it's like a simplified religion style altogether. You can change different colleges and like other dining halls, their events. It's really cool. Yeah, right. Yeah, I mean, but the thing is, he's an aviator, so it's an aviator, and it's like some of the things that he wants to do, because he wants to go for his job. Like, the more that I teach, the more like I prefer research. Myself.
So if you want to run just this.
Yeah, no, like a wholly from scratch.
Oh, okay, and you think?
Yeah.
Go here. Say new. And then you have a brand new one, clean with nothing else. You can import anything, run. All right, we're gonna restart in like 2 minutes, okay? Men's room is 1 floor below or 1 floor above. I know. My lab is right here.
be so crazy because you know it's never computed up. They're actually doing it
So Jim has so it's still an array, right?
Right. Well, the thing is, like, it used to be sort of just, you know, if it comes down to it, we do have to be that crazy. Then like, so, let's do a lot of that opportunity. And you know, you don't do quite as much in your life.
Yeah, so still is very high level in terms of how treats images. So it kind of moves. Look, like when you take this object and just run it, or the points these are only different things like implemented. So this has a lot of layers between you and the image instead of numbers. So we'll list of
So like, I just want to finish it, just want to finish it. But then I just want to finish it.
not for what but you can like look it up to see whether like what you can use cyclic image there on the image or you can right. So we're getting into more I guess complex or interesting parts of image analysis, right? So far we're talking about what is an image, right? A bunch of pixels, and then we're talking about pixel levels operation. So you take each pixel, you do something to it, and you put it back where you found it, right? So now we're going to look at environments. So instead of looking at this one pixel, do something and put it back, I'm going to look at this one pixel and some pixels around it. I can pick whatever environment I want, do some calculation based on this environment, and put this pixel back where I found it, right? Does that make sense? Okay. This process that I just described is called spatial filtering, right? And the idea behind spatial filtering is that I create a certain filter. So a spatial filter is just a small image, basically. And that small image has, like any other image, pixel values for any position. Usually, that image is called a kernel. right? So the kernel is the small image that I'm going to take and kind of apply to my big image. Now this kernel has weights. These weights are just numbers, right? So I'm going to say, you know, for this 3 by 3 kernel, I'm going to have weights W1, W2, so on and so forth. Now in each position here, so here, I'm going to go and take this kernel and just kind of slap it on my image and just multiply position by position. So I'm going to say W1 times this pixel, W2 times this pixel, so on and so forth. And the outcome of everything, I'm just going to put back into the center pixel. So the center of my kernel is what's going to be receiving the output. Right? Now I'm going to do this to each position. Okay, and I'm going to create a new image, which is the filtered image, basically the result of all these calculations. Okay, so the concept makes sense. Okay, now this kernel can be of any shape, any size, right? It doesn't have to be square. It can be like oblong. It can be circle. It could be whatever. It could be big or small, depending on how much information from how far the neighbors you want to put into this 1 pixel. Sometimes you care only about the next nearest neighbors. Sometimes you care about stuff that are like 100 pixels away. Sometimes, you know, anything in between. This procedure is formally convolution. So if you have any kind of math background, so this procedure of going to every position, doing some operation to all the environment of that position, and then putting it back to that position, formally this is called a convolution, which also I call it here filtering in the spatial domain, but this also makes the connection, if anyone has heard of Fourier filtering, these things are related, right? So this is not separate. A convolution in physical space and image space is multiplication of just 2 matrices in Fourier space. So I'm not going to go into these details, and I'll be more than happy to discuss them in private, but this is just kind of, these things are often kind of mentioned as 2 different things, but in reality, they are the same thing, just applied in different ways. Okay, so this is kind of the idea. So you see how here I take this window out of an image, or sorry, this filter. I take this window out of an image, and just this calculation will give me a different number here, and that's the number I'm going to get. This specific filter, you have an idea of what it does. So can you see the numbers? It's like 1/9 over all of them, and this is a 3 by 3 filter. So what exactly is this filter doing? What happens when I add up all these pixels and then divide it by 9? So I average, right? So this is the local average of the image. So when I apply this filter, what I'm doing is I'm taking the local average for every position. When do you think doing a local average would be good? What would it give me? So it would smoothen things, right? So for instance, if I have a single pixel that's like, for whatever reason, my camera chip had a hot pixel, and I want to get rid of it. So doing something like that would do a lot to get rid of it, right? You can select, so this is, for instance, linear filter. We can do nonlinear filters where you select the maximum. They're slightly different because you cannot actually represent them as a convolution, right? So they are they're different in the sense that mathematically they are applied in a different way.
Like data analysis, like the maximum point is more preferred than the average.
Not necessarily, right? So it depends on what you're doing, but for instance, max pooling, if you have a hot pixel, would always keep the hot pixel. So it would always increase your noise, right? So this is, I think you're talking specifically about kind of convolutional neural networks, right? And that's a whole different story that we're going to maybe talk about at the end of the course. We will talk about at the end of the course. But for now, it's unrelated specifically to this. So this is the idea of filtering. So I know this is a little subtle. So please, if you have any questions, ask them now or as we get into implementing things. Now, the one thing that filtering is good for, I mean, filters are good for many things, but they're very versatile because right now we're talking about like local averaging or something like that. The other thing filters do is kind of pattern matching. So if I have, let's say, a circle, so I make a filter in the shape of a circle, and I go and apply it to a certain image, I will get the maximum outcome, what's called response, and points in the image that look like that circle. And so if I want to measure how circly something is, I can apply a circular filter and get as an outcome an image that gives me a score for how much like a circle every point looks like. And we're gonna do some of this as we go along. So okay, so the first thing we're gonna do is kind of simple smoothing. Okay, and the most common way to do smoothing is using Gaussian filter. So our kernel is going to be a Gaussian, and what numbers define the Gaussian? What do I need to know about a Gaussian to know? Width, right? So the width of the Gaussian would determine how far am I doing the smoothing. So am I smoothing over 100 pixels or am I smoothing over 2 pixels? Basically, just kind of like making sure my local environment is smooth. So please go and to 1.21. Also, it would be useful maybe to read everything here, although I mentioned most of this already. So smoothing, it's sometimes also called the low-pass filter. And you remove kind of the high-frequency stuff, so things that have big differences between neighboring or close cells. These are high-frequency components. So go and try and implement this. The right module is filters from skimage. So I'm already calling it. You can go in here and the link is there. And yeah, so I'm going to give you 20 minutes, I guess, to get to 1.3. So 1.3 is where we start talking about masking. And over these 20 minutes, you're going to go over both smoothing, sharpening, which honestly is not often used, and I didn't leave anything for you to do there because it's just something you might want to do to pictures of your grandma from her 80th birthday, but usually you don't want to do this to images of your data. And the last type of filter that you're going to apply is kind of edge detection. So often in data analysis, we care about finding edges. For instance, if we're looking at cells, we want to see the edges between what is dark and background and bright and foreground we care about. So please go from until 1.3 and about, let's do it in 20 minutes, okay? Does that sound reasonable? Let's do 15, because you're probably not keeping time. I can just tell you whatever. you can go and seek specific people who are contributing to this specific package, but in general, this is all community area. Figure out how to convert it or not. Yeah, so the other thing is that it's coloring, so I'm not sure exactly the structure of it. So yeah, I would have to look at it. Yeah, because that's object is not. Yeah, so. Yeah. Yeah, it would need to like convert it to. So you would need to do some hard converting from one of those structures. Again, the problem is so this is the what I just told you, better community. That's also sometimes the downside because the different group is putting the one thing and another thing, and no one ever offers these two things all together. So you kind of have to figure out how to convert it from there. Oh, that's a good.
Oh, really?
Sure. The step is just pixel. So, you know what JPEG is and how what it does exactly? So the way JPEG is it's the way it's nested just values that correspond to labels and different scales in the data. And most of them are going to be zero because it throws away a lot of labels. And so this is how it does compression. Instead of keeping big matrix numbers, it's a list of numbers, and the way we use the file format is standard. Each one of these is then converted to filter. Kind of very similar. So this is not as easy to work with, and if it's just points of page with color. The other is different. That's probably the primary difference. So we can probably open it. Sorry. What are sort of the what do you do? Okay. They're ophthalmologists? Yes. Okay. How was my explanation about your eye?
That was very good.
Okay. Yeah, exactly. Yeah. This is the spherical aberration, common aberration. They're going through specific. It's the same thing.
Yes. So we have these matrices that are imaging that. To make this smooth, we use the same like would be have them as representing the matrices.
Yeah.
So if you use these commands,
Yeah.
Yeah.
So probably it depends if you have if there are multi-channel, so you would have to apply it to every channel.
Yes.
And I think the Gaussian, this specific filter, the Gaussian, it's actually already implemented to take multi-channel images, that's fine. But if you want to use investment filter, then you just have to have every channel. So what are the images? Oh, the OCT. Okay, so you shine light.
We are not changing.
Okay. Okay.
So the information of one machine and we are changing.
So how are you doing this?
We're doing it on. With on, like the machine is on to the systems that we
So I'm saying so let's say the first one gives you like a 500, 500, and the other one gives you like 200, like
the first one has 200 or 200, 760.
So you want to take a 700 whatever and make it smaller.
Very small. Average.
So so you average it yourself, or do you do some like anti-aliasing or something?
First, we do that. Very normalize, and then we average it.
Okay.
Just like because we say that okay, these degrees are these amount of this. We want to change by 25 degree in between. So average, so that amount of
Okay. And the problem is what comes out on the other side looks like blur.
yes, the one that's 760 to 61 small, it's not that very nice. It still uses a videos. Yeah.
Okay. This is probably the problem with not having enough anti-aliasing.
Yes.
So what happens is like you're averaging, you take like these 10 pixels from here, whatever, 10 pixels from here, 10 pixels from here.
Yes.
And you don't do anything before or after to make sure that they're really smooth. And just for whatever reason, this is going to give you 15 pixel value, and this is going to give you 7.
Yes.
And so it's going to look like green. It's going to have a big jump.
Yes, exactly.
So this is what you do. So the whole idea of like anti-aliasing is just so when you do the smoothness like dimensional reduction, you need to first make sure that the values in between are going to be that you're not going to get these big jumps.
Oh.
The way to do it is there's another processing step after you introduce your. So look up anti-aliasing and see how it's used. Oh, I'm sure. So you're going all the way until 1.3, right? So you're doing smoothing, sharpening, and edge detection. So the sharpening one, even though there is nothing to do there in terms of coding, do look at it and see like what exactly is going on, right? Like go over the code, try and understand what's happening. You are? Oh, that's terrible. Ah, shit. We have a minor crisis, so 1 second. What did it say? Right. How's it going? Did you manage to smoothen things? Did you manage to find edges of things? What? Edges what? You couldn't. Okay. Okay, so we're going to go over it together. Okay, so as always. So first thing we wanted to do is to just do some Gaussian filtering. It's going to come up eventually. So Gaussian filtering is, we're going to use filters.Gaussian. Cool. And we're going to filter our image, this one. Image hist. We're going to say sigma equals sigma, right? And we have a multi-channel image, right? So we probably want to set multi-channel equal true. Although it's possible that this is not strictly speaking necessary. Did anyone run this without this argument? Did it run fine? It ran fine? Okay, so let's not make things extra complicated for ourselves. And, okay. So it knew that this is multi-channel, and it told us that if you really want to tell it that it's not multi-channel, make sure to tell me that it's not multi-channel. So you see this is the original image, and this is the smooth image. Did anyone play around with changing the value of sigma and seeing what that does? So let's try and do that. What would happen if I put sigma at 0.5? What do you think? How would this image change? Less smooth, right? It'll look sharper. And if I put it at 5, then what would happen? Be blurrier, right? So let's look at that. Blurrier, right? Cool. Now the next thing is sharpening. Like I said, sharpening is not usually something we do. And you see it can have like limited effect. The way sharpening is implemented here is we take our our image that we want to sharpen, right? So usually we want to start off with a kind of crappy image and then make it better. We're going to do a Gaussian filtering to that with a small Gaussian and just remove that image from our input image. So the idea is I'm going to remove all the low frequency components, and whatever is left is going to be relatively high frequency. So for what I have relatively high frequency, I'm just going to add that back to my input image with some factor. So I can play around with that factor to have more or less sharpening. Now, again, the garbage in, garbage out is always true. So this is not magic. We can't create information where it doesn't exist, but we can make it look slightly better. So if this is the input image, this might look slightly better than what we started with, okay? And then the final thing, which is actually a very important filter to use in most analyses, is the idea of edge enhancement. So most or all edge detection filters are shaped, or if you take these values and kind of shape them, like kind of plot them as a curve. So what it usually looks like is something like this. So if this is the center of the filter, there's going to be some negative number on one side of the center and some positive number on the other side of the center. The idea being that if I apply this to something that is flat, then I'm going to have negative, let's say on this point, I'm going to have the negative value times this plus the positive value times this. And they're going to give me 0, right? So negative plus positive times a constant gives me 0. But if I have an edge, something that looks like this, then I'm going to have the negative value times this plus the positive value times this. And this is going to give me presumably a large number, right? And so this kind of goes back to the idea of filters being kind of detectors of different features, right? So this detects the feature of an abrupt change, a large gradient, right? And the response is going to be a large number wherever I have a large change in intensity. Now there are different types of edge detecting filters that you can use. Each one has its own name, but they're relatively simple. Now notice that in this case, you need to have one filter that detects edges in one, like the horizontal axis, and one that detects the edges in the vertical axis. So this is true for Sobel. So this is just the name of a guy, and Pruitt. And then you have one called Roberts that detects gradients in like diagonal directions. If you combine the two directions, so you take the magnitude of the gradient, combine it in both directions, you can get the total amount, the kind of total gradient in that position. And this is how you can kind of detect edges that go in this way, detect edges that go in this way, and then detect total edges everywhere. Now, let's just implement this, and then I will have another kind of small comment on this idea. So we're going to use Sobel, right? So filters dot Sobel, and we said we want just the magnitude here. So this is just Sobel, and this is going to be of, and I'm going to apply it to just one of the RGB things. The dimensions. I think you should. This should have. So now, to apply it to the horizontal direction, I'm just going to call Sobel H, and to apply it to the vertical direction, I'm just going to call Sobel V. And what we end up. Sure. Yeah. So this is just so I don't need to deal with RGB, because this is implemented for just one channel. But in general, if you want to apply it to RGB, then you just apply it to each one of the dimensions separately, right? So it's nothing different. This is just an implementation thing. What? Again? Here? What? I don't understand.
Okay. So what I'm saying is that, so you already got filter image in the first line, and the second line we just get a filter image in the brackets instead of
what? Okay. Okay. So this is going to be the magnitude. So this is going to kind of give you all the edges, no matter in which direction. This will give you edges that are horizontal, so things that are kind of lines that are across the image. This is going to give you edges that are vertical, so lines that are up and down. Now, there are some neat modern-ish implementations of filters that are steerable. So the idea is that you can, in one go, get both the magnitude of the gradient and the direction of the gradient. So if you look up on Google something called a steerable detector, so what it does is that it more or less, in a slightly more complicated way, applies this edge filter in all different angles, and it returns the angle where you had the highest response, and the amplitude of that response. And that way you can get a better idea of like exactly to which direction something is pointed. So for instance, if you're doing anything that has like microvilli or something that microtubules, whatever, you can analyze both the direction and the existence of these microtubules in one go. So this is just something you can look at later on, and this goes under steerable detectors. Steerable, so you can it steers like. So I don't think it exists yet in like scikit, whatever, but there are Python implementations of it. So if you just Google it, you're going to find someone who made it. I think the paper that first described it was like 2014. Okay. So any more questions about kind of this idea? So it's a combination, but a magnitude combination, right? So it's so the magnitude, so let's call it. So magnitude, right? So this is like Pythagorean magnitude. Okay. Okay. So also you see that like we have these like implementations like Sobel, like you can just call this, but also implementing these things just by calling it by filtering isn't too hard, right? Because these are not particularly difficult filters to implement on your own. Usually we don't need it, but if in any case you come up with your own idea of a filter, for instance, I don't know, one like the ridge, like something that has like just a line, right? So 0, 1, 0. Right? Maybe that's what the feature you care about. So you can implement whatever filter you want and just kind of multiply it or do a convolution between it. And that's something that does exist in NumPy, between that filter and whatever image you want, and it would give you a response to that filter in any position. Okay, so we're getting there. So the next thing we want to do is something called masking. Okay, so this is something I started talking about early today when I was still talking about kind of what is image processing. And masking in its kind of simplest form is the idea of in any image, or mostly in most images, I have some part of the image that I care about, that I usually call the foreground, and some part of the image that I don't really care about, that I usually call the background. And masking is the idea of separating these two things, right? So how do we do that, right? So is that clear? Kind of what's the goal? Okay, so a mask. What a mask is a binary image, and by a binary image I mean an image where every pixel gets the value of either 0 or 1. It gets 0, 1 if this is a pixel that I want to keep. It's a pixel of my foreground. And it gets the value 0 if it's a pixel in the background, something I don't care about. And the way you apply a mask, so we call it applying a mask to an image, is just by pixel level multiplication, right? So I take pixel number 1, 1 in the mask and multiply it by pixel number 1, 1 in the image. And if it's 0, I'm going to get 0. And if it's 1, I'm going to get whatever the value in the image is. So is that clear? Yes, no, kind of. We're gonna do that a bunch, so it will become clear. And so we're going to start off by kind of like nothing that's motivated by the data itself. We're just going to make a mask of our own desire. So we can always do that. We can always say, I'm going to make a mask and I'm going to take just this region of interest. So this is another thing masks can be used for. You can define your own region of interest. You're going to say, I don't care about anything that happened in all this part of the image. I really care about this circular bit in the middle. I'm going to take that circular bit or any bit, and that's the only part I'm going to focus on. So you can create your own mask, and by that you eliminate everything else. So that's exactly where we're going to start this. So we're going to start by just making a circular mask. And we're going to make that circular mask, we're going to make a circle of radius, whatever it is, 100. So I wrote it somewhere. And I'm going to make a mask where everything closer than 100 is 1. And everything above 100 is 0. So I predefined the size of my total mask. It's 512 by 512. You can see it here. Okay. And the first thing I need to do is to define my kind of distance from the center. Do you guys want to do it, or do you want me to show to do it here? Show, because this is kind of like, this is math. So I'm going to show this part. It's not particularly complicated, and then I'm going to let you do the rest of this kind of yourself. Okay, so how do we make something that is just the distance of every point from one center point? So I already defined the center points, center y and center x, and I have these positions y, y, and x, x, which are just every pixel gets its own value. So radial distance, we know it's square root. Just wrote this. So np dot square root of what? So it's going to be xx minus cent x. And this whole thing we're going to need to square. So this is how we do powers in Python. Right? So it's not this thing, it's the 2 stars. And so this is the distance from x. This is the distance from y. I probably have some open bracket. And so this would just give me, for every point in this array, what is its distance from the center. So let's see that this is what we got. So in the very center, we said black is 0. White is a large number. And what we get is that it's very small in the middle, and it grows towards the edges, right? So this is not surprising. This is the radial distance of every position from the center of this image. Cool? Okay. So now we're gonna go into masking, and I'm gonna give you 15 minutes to get from here to 1.4. All right? Or, okay, we don't have 15 minutes, evidently. So we're going to get to You guys want to start over from here tomorrow? Yeah? We'll just stop here. We don't want to cram. We want to do it there. Cool. So you can say it automatically saves. You can also save it. There is like a little save button here. And we're just going to start tomorrow from 1.3 and hopefully get somewhere. All right? Cool. Questions, answers, Andre's comments.
Can we talk about that image
HGF5. Let's not bore everyone with it, but we can talk about it. Okay, so that group of ophthalmologists, we have a strong ophthalmologist presence in the room. So we can sure. Yeah. All right, so see you guys tomorrow. All right. Thanks for your time. Yeah.
There was a part at the beginning, I'm just trying to tell me that code is doing. I have a GCA or something. It was one of these.
So GCA means get current access.
Yes, I know what that means, but oh, here. Okay, and then it says subtitle, so it's like giving the title, right?
I'm just setting the title for this.
Right. But why is this code important? Because when I run it without it, it won't work. That's what I'm confused about. Because it seems like it should be something that's not important, right? Oh, it works.
Yeah, it works. Just
But on
without.
one of these, I tried it and it doesn't work. I'm just trying to understand. I don't know, I can't find it anymore. Or maybe it's here. No.
So it should generally work. The only
Okay.
reason I can think of maybe it not working is if I make a title and then I hold that title for whatever reason. And if I call the title for whatever reason, I change its position or whatever, and it's not there,
Okay.
and it's going to cost a few.
Okay. If I find it, I'll let you. Okay.